No. Oh, there's two in our physics class, Patrick and Colin. They sit in the front row. And then there was Naya Somerville.
All right, that clock is a little bit off back there, so I'm going to go by the computer. Says it's 1:30. So let's get going. You did have an exam that was just last Wednesday. I know it maybe seems like a long time ago. I'll talk about a couple questions that were commonly missed next time, but I can tell you that the mean was a 76. So it was higher than what I expected. I did have one of you that got 100%. So overall, not bad. Come see me if you want to look at that. Like I said, we'll just go through a couple questions next time that were commonly missed. There was one slide of staff that I didn't get to. I'm just going to forget about that slide. It was just kind of a couple other staffs that are not as interesting as Staff Aureus. So you can just kind of cross that last slide off. Let's move on. Let's talk about the final exam. So 2 weeks from today, hopefully the weather will get worse again. So you have a little better studying weather, right? This week is supposed to be too nice. So next Monday, note the time, come an hour and a half earlier, right? Come at noon. So 12 to 1:50 in this room. So just to give you a little idea of what that will look like. It's approximately a third of the exam that will be new material. So starting with the vaccines, which wasn't covered on exam 4, and then Staphylococcus that we finished on Wednesday after the exam. Into today, we're going to talk about Streptococcus. We're getting into the pathogens. Okay, so it'll be, I haven't finalized the exam yet, somewhere in the order of 45 questions. So what pathogens are we going to cover? I'll come back to that slide in a second. This is kind of a list. I talk about kind of different ones every semester. I think already looking ahead, we're running a little bit behind schedule, and I think that we're not going to do tetanus or botulism or whooping cough. I can kind of confidently say, you know, we gotta trim a little bit. I think we won't do those. So that follows the exam objectives that are posted. Nothing with tetanus or botulism. I'll post a updated exam objectives once I'm a little bit more sure what we're going to get to and what we won't. If I have to cut something else, I'll cut the smallpox and the virus, but let's wait and see how we're doing. All right, so those are the pathogens that I'm kind of anticipating getting to. Most people like this part of the semester when we kind of focus on different pathogens. What are we thinking about those pathogens? Their morphology. So that means, is it a gram-positive cocci, for example? Is it an RNA virus, for example? What kind of diseases do we get from them? So, you know, especially interesting signs and symptoms that we experience. What virulence factors can the organism make that makes it successful in causing those diseases? So that's probably the part that will be a little bit less familiar, that you have to spend a little bit more time maybe looking at those, although some of them we talked about before, and we're saying, here's an example of that in use. In some cases, I've gotten really specific in talking about treatment. In other cases, it's, you know, antibiotics, a lot of different antibiotics. But again, just depends how specific we get. For some of these pathogens, we have a vaccine. So if we do, what's that vaccine called and what kind of vaccine is it? Okay, so I have some other more specific objectives written, you know, in your big objective list, but those are kind of the main things to kind of think through and the bases that I'm trying to touch on. All right, so that's the new material. I encourage students to spend most of their time studying this new material because you are going to see questions on that. You're going to get the most bang for your study time studying those new pathogens. I hope that you will also be able to take some time to look back through the semester although there's less chance that you're going to see, you might, study, for, I don't want you to study hours and hours and hours on metabolism, and then you only see maybe 3 questions on metabolism. So, or maybe less than that. So kind of think through that. Definitely do the new material first. What does the cumulative part look like? Again, this is about approximately 2/3 of the exam. The multiple choice part is the smallest part, and it's also generally the more difficult. I try to take maybe 7 questions from exam 1, 7 questions from exam 2, not the same questions, but from that material. So that's the part that's a little bit harder to recall. Although I will say I try to ask questions about things that maybe we've talked about several different times. So we talked about the structure maybe back in chapter 3, and then why that was important later on, and maybe the immunology of it. So I try to come up with questions like that. The matching part is the part that students do well on. So, all these terms, of course, we've talked about a lot of different terms, but you really only need to be kind of familiar with them rather than kind of being able to spit out a definition. All right, so I'm going to show you an example of that in a minute. I will say a few hints on the cumulative portion. I won't give you any questions on history, so you can kind of, not forget that part, but not emphasize that. I won't make you memorize all those specific antibiotics again. So those list of like 14 different antibiotics that you had to know the mechanism and, whatever side effects and things like that. You don't need to go back through those. Or the chemicals. There was like 5 or so chemicals. So I could still ask questions like, what does it mean for an antibiotic to be broad spectrum? Or what is the therapeutic window, therapeutic dose refer to? That sort of thing, but not specific antibiotics. So I don't know if it's any help or not, but these are sort of the areas that I consider particularly important, and I know that that's covering most of what we talked about, but I just tried to give you a little bit of a, way to kind of think about what you're studying. So, if there's one of those tests that you did a little bit poorly on, then you might especially want to review that material. Okay, the matching. So I think typically what I've done is had 4 different pages of matching. So that means there's somewhere between 12 to 15 terms on a page. So here's an example. I had, I thought up a whole bunch of terms, and these are some that just didn't make it onto the test. So there's 12 different terms, and I had 13 different descriptors. So there may be one extra descriptor. I think on one page, I've usually done it 12 descriptors per 12 questions or 12 terms. So there may be an extra one, there may not be. For some of these terms, like mycoses, okay, that's a fungal disease, okay? You're not likely to get that mixed up if you kind of have the barest minimum idea of what that is with another question. Some of them, the difference between cell-mediated immunity and humoral immunity. Okay, yeah, then you gotta, if there's another closely related term, or do you know the difference between the TCA cycle and Embden-Meyerhoff glycolysis pathway? So in some cases, again, hopefully it's kind of a give me. Other cases, you gotta kind of know the difference. So, right, so kind of look that over. I've tried to boldface the terms in your learning objectives, so that might be somewhere to start. Anything that's bolded, not only that list of things, but that might be somewhere to start. Okay, questions about the final? All right, ask as they come to you. All right, so we talked about Streptococcus after the exam. We finished up Staphylococcus. The other gram-positive cocci, that's kind of the other famous gram-positive cocci, are the Streptococcus. And again, if you're in lab, this should sound kind of familiar. we're going to focus in on one particular organism, but in general, there's about 50 different species in that genus Streptococcus. So again, there's not a lot we're not really going to talk about, but when you gram stain these organisms, they're going to be purple when you gram stain them, and especially when you take your sample from a broth, they tend to chain up quite nicely. So the older they are, the longer chains that there will be. Some of them tend to make just pairs of cocci, diplococci. But when you see chains like that, you should think, oh, I've got some kind of Streptococcus, or there actually are a couple genera that are really closely related to the Streptococcus, Enterococcus being one of them. As with all cocci, as far as I know, there's not any motile cocci. That just means they don't have flagella. Okay, so they're non-motile, they don't form spores. Maybe one spore former that's a cocci, but not the famous one. So mostly they're non-spore forming. Facultative anaerobes, there are a few that are true anaerobes. So there's a few exceptions there. Some of them are considered capnophiles. And that means they grow best in high concentration of CO2. All right, we put brackets around something that means concentration. So you should know that if you're in lab, because our Streptococcus, we either put them in an incubator that was hooked up to a CO2 tank, or we grew them in a candle jar. So we light a flame in there, and that flame produces a little CO2 until it goes out due to lack of oxygen. So again, some of them, they will grow best, or even they need that high concentration of carbon dioxide. The oxygen requirement is a little bit complicated for these organisms. Some of them, even though they don't ever respire aerobically, we still consider them facultative because some of their virulence factors are produced more often where there's oxygen present. So again, whatever, it's a little complicated, in other words. But they do not make catalase. So this is how we tell the difference. It's an easy way to tell the difference between the Staphylococcus and the Streptococcus. The staph make catalase. It's this enzyme to detoxify hydrogen peroxide, which is a reactive oxygen species that's produced as a byproduct of aerobic respiration. The streps actually produce a different enzyme that does the same thing, called peroxidase. So it detoxifies that hydrogen peroxide, but without producing any oxygen. Okay, so those of you that are in lab, okay, so when we do the catalase test, we look for the production of bubbles, which are the oxygen. Okay. The classification of Streptococcus gets a little bit confusing, more so than Staphylococcus, because we have two different ways that we like to group them together, which are don't have anything to do with each other. We like to classify them based on hemolysis patterns, and that's kind of what we've done in lab. So when we streak our strep on blood agar, they have a characteristic way that they will break down those red blood cells. If we call them alpha hemolytic if they partially break down the red blood cells. So that means they lyse red blood cells, but they don't break down the hemoglobin. So that intact hemoglobin turns the media a little bit kind of greenish brown. If they are beta hemolytic, that means they will totally lyse red blood cells, and then it looks a little bit kind of yellowish around the growth. You can miss that if you don't really hold that up to the light. If you're just kind of looking down where it's dark against the bench, you can kind of miss that. And that is enhanced where there's places where there's less oxygen. So when you streak that out, you want to stab down in the agar, and you often see even more beta hemolysis from organisms that have the ability to be beta hemolytic. And then gamma hemolytic is really no hemolysis. There's not any change in the blood agar at all. They're not lysing the red blood cells at all. Okay, so we would call that blood agar differential media because we can see the different hemolysis patterns. Another way that we will classify strep is based on Lancefield groups. And Lancefield groups, they were named by Rebecca Lancefield back in the 1920s. She discovered that a lot of Streptococcus have these carbohydrates on their surface. So we call them carbohydrate surface antigens. Usually carbohydrates aren't very antigenic because they're repeating units, but these can be identified with the use of antibodies. So it's a little bit beyond what we would do in lab here using antibodies, but you can type these out into different groups and not to memorize these groups. I don't have this memorized, but it kind of shows you some different streptococci that are out there. And also notice that this grouping system, it doesn't go strictly in order of the alphabet, like it skips some letters. So I'm not sure why they did that. And also note that there are some streptococci that don't have these carbohydrate surface antigens. So we call them non-Lancefield streptococci. And actually some of them we put in this group we call the viridins group. Viridins meaning green, so they're alpha hemolytic. And you'll notice on this that there are some lactococcus and some enterococcus. So back in the 80s, they decided this group Streptococcus was too big, and they peeled off some that they called enterococcus and some that they changed the genus name to lactococcus. So, but they're very closely related to Streptococcus, so we can kind of use some of the same tests to identify them. So for some of these groups, like group A, there's only one organism that makes up that group, Strep pyogenes. And so we have an abbreviation for Strep pyogenes that you see sometime, it's kind of a cumbersome abbreviation, but that GABHS, that stands for group A beta hemolytic strep. Okay, so again, it's a little bit cumbersome, but sometimes you see that. Some groups, like group C, there's several different species that make up that group. Okay. So again, when you see that, that just refers to, you know, some classification for Streptococcus. All right, so I want to zero in on one particular organism that's clinically interesting, and then just kind of briefly mention a couple of other Streptococcus. Like I mentioned, Strep pyogenes, it's beta hemolytic group A. Where do we find it? Mostly on people, kind of the upper respiratory tract, like the throat, the nasopharynx, sometimes on the skin as well. And these organisms are fairly wimpy in the environment. We talked about the Staphylococcus as being salt tolerant and, you know, being able to withstand temperature changes and things like that. Not so much a Streptococcus. We don't find them out in the environment, in the soil so much, or rarely. They're pretty picky about the media that they grow on. They need a really rich nutrient source like blood agar. So mostly humans, rarely animals. Let me write that in here. We'll say it can be zoonotic transmission, but rarely. There have been cases where people have got a Strep pyogenes infection from their pet, they think, but not commonly. Usually someone gets a strep infection by a respiratory secretion. So those large droplets, not droplet nuclei. If it's droplet nuclei, then that's going to be airborne. Typically, this is not airborne, just those large secretions when you're close to somebody, when they're talking or coughing within a meter. Okay, so usually you get that by being in close contact with somebody. Occasionally, we can have some Strep pyogenes just as our normal microbiota in kind of low numbers. All right. All right. Oh, I can't ignore that picture. I put that up there because this page seemed too blank, but there's this website called Giant Microbes that has little plush, you know, E. coli's or Ebola or whatever. So this is the strep pyogenes from Giant Microbe. So again, it's got, you know, diplococci there, and it's got a knife and a fork printed on here. So anybody have any guesses why you would have a knife and a fork? It's okay. Hold that thought. We're going to get to that. It's one of the infections that it causes. Okay. All right. So we'll get to that in a minute. Let's first talk about some of the virulence factors that make strep pyogenes successful, and then we'll describe some of the infections that it causes. A couple of these things we've talked about in the past, and we were talking about examples of antiphagocytic factors. So strep pyogenes will make a capsule composed of hyaluronic acid. So hyaluronic acid is something that we have. It's kind of the glue that holds our cells together. So it's something that's very similar to what hosts have already. So it's just not typically recognized as being foreign. So that helps it evade phagocytosis. As if that wasn't enough, the strep pyogenes also has this little protein on the surface of its cells called M protein. And we mentioned this before. Here's a cell with M protein, and here's a mutant that's lacking the M protein. This mutant that's lacking M protein probably wouldn't be able to establish itself in a host. It wouldn't cause any infection at all. That M protein, again, it's mostly antiphagocytic. It also will inhibit activation by the alternative complement pathway. So remember, the alternative pathway, that's the one where the C3 is kind of being split at a low level, and if C3b happens to be in the vicinity of the pathogen, it will bind to it, and then that pathogen and complement fragment will split lots more C3. There has to be some proteins that we didn't really get into also in the vicinity. The M protein actually binds to one of those proteins that's important for that activation pathway. Okay, so that not only will inhibit phagocytosis, but some other like inflammation and chemotaxis of cells. The other interesting thing about the M protein, there's different kind of variations of it on strep pyogenes. So one variation is called rheumatogenic M protein. And this is important when we think about some autoimmune complications that strep pyogenes can cause. That rheumatogenic M protein can actually cause our antibodies to cross-react with the host heart valves. Okay, so antibodies that we make to target that rheumatogenic M protein can also kind of target ourself, cause heart valve damage. And the nephratogenic M protein, their antibodies produced can cross-react with kidney antigens. Okay, so we'll talk more about that as we get into disease, but that's why you want to treat strep infections so you don't hopefully have to deal with those autoimmune reactions, complications. Other virulence factors, a couple more than this. I just picked out some question. Oh, yeah, did I go too fast there? Okay, another question. It inhibits the complement pathway activation, but it's probably, and that is going to play a role in inhibiting phagocytosis as well, because you don't get the opsonization, at least by that particular complement activation. Okay. All right, good. So some exoenzymes, again, I just picked out some virulence factors. There's other ones, especially ones that have ties to something that we talked about. So C5a peptidase, if you remember, C5a is a important factor to increase inflammation, to increase chemotaxis of other white blood cells to come and help fight the infection. Okay, so this is an exoenzyme that neutralizes C5a, which we'll call a chemotactic factor. Okay, so decreases the number of white blood cells that are going to be called to the site of infection to help fight the infection. Another exoenzyme that strep pyogeny produces is called streptokinase. That's going to be important for, we'll say, important for invasion. Say invasion of tissues. If there's, say, a wound that the bacteria are infecting and the host blood has started to clot a little bit, it's going to be able to break through those clots in order to invade deeper into tissues. Okay, so it's spread of infection, invasion of tissues. Another one kind of reviews back to the function of neutrophils. Remember, our neutrophils act by phagocytosis, degranulation, and the third way are those neutrophil extracellular traps. So this exoenzyme streptodornase is actually a DNase, so it breaks down DNA. So the bacteria, when it secretes that, if it's caught in one of those neutrophil extracellular traps, where the neutrophil has kind of expunged its DNA to immobilize a pathogen, then that strep pyogenes can break out of that trap by being able to break down that DNA. All right, so again, it's an arms race. Like, we do something to fight it and it has something to fight back. A couple of interesting exotoxins. Remember, no endotoxins. It's gram-positive pathogen. The streps, like we talked about, will make hemolysins, characteristic ways that it breaks down red blood cells. So we call the hemolysins that streps produce a streptolysin. And they actually will not only act on red blood cells, but they actually act on other formed elements of the blood, like white blood cells and platelets as well. Obvious rule, there's a lot of nutrients in red blood cells, so acquire nutrients that way. And this toxin plays kind of a dirty trick. If the organism is phagocytized, it can release this streptolysin that causes the lysosomes to degranulate before it fuses with the phagosome. So all those digestive enzymes will kind of act on the phagocytic cell and can bring about the death of the phagocytic cell rather than the death of the pathogen. Another exotoxin that only some strains of strep pyogenes will produce is an erythrogenic toxin. It's a super antigen carried on a lysogenic phage. All right, so let's review. Remember, super antigens cause massive T cell activation. So massive amount of cytokines release, lots of inflammation, and reviewing back to our viral replication, remember a bacteriophage that does lysogenic replication. That means that it will incorporate the viral chromosome into a bacteria cell. Well, sometimes that will change the phenotype of the bacteria cell. Okay, so in this case, the bacterial cell, the strep pyogenes, if that's been infected by a particular bacteriophage that's carrying the information for this toxin, then the organism that's when the organism can make this erythrogenic toxin. Okay, so let's see. So if it's carried by that transduction, remember that's the three kinds of horizontal gene transfer. We have transformation, conjugation, transduction. Transduction was the one where a virus is carrying a gene and it will kind of accidentally carry it into a new cell. Okay, so it's an example of lysogenic conversion, where no T, conversion. Okay. Lysogenic conversion. Okay, so organisms that, the strep pyogenes that can make that erythrogenic toxin, that causes them to make this bright red rash when they're infecting us, and that's an infection known as scarlet fever. So again, it's only strains that have been infected by a virus that has given them the ability to make this particular toxin. And strains that have been infected are not circulating as commonly now. So that's not a very common infection, but we'll talk more about that in a minute. Okay, so if we start thinking about some different infections, and again, it's different strains will cause different infections. It depends on what virulence factors that strains have. We start with some skin infections. Impetigo is a good place to start. Remember, we talked about how Staph aureus can cause impetigo as well. So it's a very similar looking infection. Sometimes they'll say, oh, strep pyogenes impetigo looks a little bit crustier, the lesions, than what Staph aureus does, but looks very similar. Remember, this is an infection that stays localized. So it takes a long time to heal up. Oftentimes it will scar if we don't get some antibiotics on there, but it's not something that's otherwise dangerous other than maybe causing a little scarring on the skin. Pretty common. Getting a little bit more invasive, a little bit more dangerous is an infection called erysipelas. Sometimes this is described, somebody has skin with an orange peel consistency. So the skin gets kind of tough looking and, you know, it looks bright red. It gets a little pitted, kind of like an orange peel. Patients usually have a high fever associated with that. It's not just the skin. Sometimes they're vomiting as well. And there are a lot of other complications that can happen: vomiting, fatigue. Oftentimes somebody can contract erysipelas if they have some kind of mild trauma to the skin. So like a little cut, could be a burn that the pathogen, the strep pyogenes, can have access then. And this can be mild and not such a big deal, or it can be life-threatening. So again, there's a lot of complications associated with it. So it's a pretty serious disease, can be. One that's often, sometimes it's kind of hard to tell the difference between erysipelas and cellulitis. They're very similar infections. Cellulitis often also, same as erysipelas, face, hands, legs. That group A strep pyogenes, again, commonly enters through some small cut in the skin or somewhere where that skin has have been irritated a little bit. Usually localized inflammation. So you can see this hand is swelled up, it's red. There's some fever associated with that, swelling. Again, it can spread to the bloodstream and also become life-threatening. So I guess the take home is if either erysipelas or cellulitis is suspected, you go to the doctor and you get that checked out and you go on some antibiotics. Okay. And then this, I think with this citation, I think there was a little bit more information information about that if you're interested. And then getting to much worse, erysipelas and cellulitis can both turn into necrotizing fasciitis. This is the flesh-eating disease. So that's why the fork and the knife are on the little flesh strep pyogenes, flesh eating. And there are other organisms that can cause necrotizing fasciitis. Of course, this is particularly nasty. If it's something as necrotic, that means death of tissue. The fascia is just connective tissue that kind of keeps all your skin and organs and everything all together. Again, this is rare, but it's certain strains of strep pyogenes that have these exoenzymes that will just digest that fascia, that kind of connective tissue, and it reproduces and spreads very, very quickly. It's treatable if you catch it early on. The problem is early on, you don't know that you have a life-threatening infection. It's, the prodromal phase is, you've got maybe a little fever, a little redness of the skin, maybe it hurts a little bit, and then kind of bam, it turns into, something that looks like this. Mortality about 50%. So we can treat that with antibiotics, but the antibiotics don't do a lot of good because blood flow is restricted to areas. So it's hard to get the antibiotics where they will do good. We typically have to remove the infected tissue surgically and try to stay just ahead of it. Okay, and again, here's a website that has a little bit more information about it if you're interested in knowing more in that. Okay. More strep pyogenes infections. There's a lot of them. This is one that probably you've all had at some point or another. Streptococcal pharyngitis, also known as strep throat. Now, most of the time when we have a sore throat, it's not due to strep pyogenes. Most of the time it's viral. But if you have some of these symptoms, it's worth checking out. And now you don't have to go to the doctor. You can go to Meyer and get a quick strep test, and they will treat you with antibiotics if that strep test comes back positive. There are some false positives associated with that because some people do have strep pyogenes as part of their normal microbiota. But if you've got some of these other symptoms, then it's worth treating. So what are some of the other symptoms and how's it transmitted? Again, droplets, it's not airborne. It's usually in close contact with somebody else. Pyogenic toxin, that prefix pyo of strep pyogenes, that means pus producing. So this is where sometimes we see the pus pockets on the tonsils. That's not always present though. So sometimes, the doctor always look back in your throat, and if they see those, then that's kind of a giveaway, but that's not always present. Fever, sore throat, really, really painful sore throat. Especially sometimes with kids, they'll have abdominal pain or even nausea and vomiting. That doesn't show up as often with adults, though. Lymph nodes are swollen, and secondary infections are common. So things like bronchitis or pneumonia, or if you have a strain that can produce that erythrogenic toxin, it can turn into scarlet fever. So again, if you think you might have strep throat, you shouldn't just, gargle with vinegar or something like that you read on Facebook. You should definitely go and see if you have it, especially again, if you have a fever for a couple days and a really sore throat, have it checked out. All of our Streptococcus are we don't have resistance to antibiotics yet for strep infections, so it's very treatable. And again, get on some antibiotics to get rid of it before you have some kind of complication, again, pneumonia, bronchitis, or scarlet fever. What are the hallmarks of scarlet fever? The rash, and this usually shows up a number of days after a strep infection. That's my daughter before she was verbal and could tell us that her throat was sore. We knew she was sick, and then a few days into it, she had this rash that looked just like that, and it was kind of a, oh, I think we need to take you to the doctor moment. And she did have scarlet fever. She got on antibiotics, and she was fine in a few days. But before we had antibiotics to treat this, this was a very serious infection. My mother-in-law, whole family was quarantined for 6 weeks when she came down with strep pyogenes with scarlet fever in the 30s before we had antibiotics. And this was the infection that they think took Helen Keller's sights and hearing. So there were lots of complications associated with it. Strains that did cause scarlet fever were much more prevalent in the 1800s, early 1900s. So for whatever reason, they're not circulating as much today. So that's a good thing. That bright red strawberry tongue is another indicator that somebody might have scarlet fever. Scarlet fever can then turn into an immune complication, and again, it doesn't have to pass through scarlet fever to get to rheumatic fever, but it can just go from having a strep not treated or incompletely partially treated strep throat. So what I mean by partially treated, if somebody maybe takes a few days of antibiotics and then quits taking them because they feel better. So rheumatic fever can come about, a number of weeks, typically after somebody has strep throat. It's most common in children. Sometimes you see these kind of bumps or nodules under the skin. Apparently that's not very common, maybe only like 2% of cases. So I didn't show a picture of that. Sometimes there's a really interesting rash that has a specific name, that a kind of rash that I didn't write down, but sometimes we see that. The heart is not, functioning quite like it should, an abnormal electrocardiogram, because again, it's autoimmune. It's an antibody reaction that kind of cross-reacts between that M protein on the strep pyogenes, and typically it's the heart valves that get targeted. Okay, so that can cause some permanent damage to the heart valves. So again, before we had antibiotics to treat strep throat and scarlet fever, this was, maybe 1 in 100 cases would turn into rheumatic fever. So you would know somebody that had maybe some permanent heart valve damage, usually the mitral valve. Okay. So again, if we ever had our antibiotics not working very well to Streptococcus anymore, then we would probably see more of this. The other possible complication is with the kidneys, where of strep throat. And there, a few years ago, there was one of my colleagues. Every time he got strep throat, he would land in the ER because his kidneys would start to shut down. Again, usually it's if you don't treat a strep throat infection, or again, only partially treat it, but for whatever reason, his antibodies that he made would obstruct the blood flow in his kidneys, and that would cause high blood pressure and urine output to go down. And the damage to the kidneys has the potential to be permanent. So again, a lot of complications. What do we do if we have one of these immune complications? They will treat with antibiotics just in case that original infection wasn't resolved. But because it's autoimmune, that's not necessarily, it's not going to dampen our antibody production. So they giving anti-inflammatories medication like ibuprofen is kind of the best thing that we can do, and hopefully it resolves itself. Okay. Any other questions about strep pyogenes? Okay. A few comments on a couple other streps, kind of the more famous ones. Strep agalactiae, that's one Some of you may have that as your unknown in the lab. It's another beta hemolytic strep. It's the only one that's group B strep. It does form capsules, and that capsule is actually a T-independent antigen. Okay, so remember that means that that capsule when B cell receptors recognize that capsule, that can activate the B cells to become plasma cells and make antibodies. It doesn't need a bunch of other stimulation, especially doesn't need any stimulation from an activated T helper cells. So remember those T-independent antigens, young children don't have good response. They don't have good B cell activation to those. So, you know, this is a pathogen that normally doesn't cause much problem in adults. It's normal microbiota, kind of a lot of different places. Vaginally is where it causes the problem, because if somebody's a carrier for strep B, for strep digalactiae, when a newborn passes through the vagina, then it can get some very serious infections like pneumonia, meningitis, or sepsis. So if a pregnant woman has good prenatal care, they will have a group B test, usually late in their second trimester. And if they're a carrier, they're given some antibiotics to try to get rid of it. And that has dramatically decreased the incidence of serious neonatal infections. Okay, and then we could talk a lot more about strep pneumoniae. I'm not going to get into virulence factors here, but strep pneumoniae is probably the other one that causes some pretty serious infections. It's a really thick capsule producer. So if you remember when we talked about horizontal gene transfer and Frederick Griffith's experiment with transformation, he used a strain of strep pneumoniae that formed capsules. That was the one that killed the mice, and the mutant strain that didn't make capsules and that didn't cause any infection at all. So a really important virulence factor, I guess I'll mention that one, forms kind of great big large mucoid looking colonies on the plate. Some infections that it causes: pneumonia, not a stretch when you look at the name, Streptococcus pneumoniae. It causes, we usually refer to it as pneumococcal pneumonia. A lot of different organisms can cause pneumonia. Klebsiella pneumoniae, for instance. We've talked about how Staph aureus can cause pneumonia. So pneumococcal pneumonia is probably the most common form of pneumonia. This organism used to be called, the genus was Pneumococcus, so that's why we call it pneumococcal pneumonia. Causes a lot of edema in the lungs. So a doctor, when they listen with their stethoscope, can hear kind of a crackling noise from all that fluid buildup in the lungs. Sometimes the patient is cyanotic, meaning the tips of their fingers are starting to turn a little bit blue because they're not getting good oxygen exchange in their lungs. It is pretty treatable. Usually patients will go on maybe a couple different antibiotics that work a little different way just to try to knock it out. But again, streptococci still respond pretty well to antibiotics. Ear infections are also caused by strep pneumoniae, especially common in young children because their ear canal is narrow and the fluid doesn't drain as easily through there. Often, start out as a cold or some other viral infection, and they'll just where again they have some fluid buildup, and then they get ear infection from that. And meningitis can also be caused by strep pneumoniae. There's lots of other organisms that can cause meningitis, but this is an inflammation of the meninges, that kind of barrier that's surrounding our brain and spinal cord. a very serious infection that patients that if they do survive, sometimes will have some permanent damage like retardation, blindness, or deafness. The good news is that it's much less common now because we have a pneumococcal vaccine. And we talked about this one before last week. It's a conjugate vaccine. So remember, a conjugate vaccine combines something that's very weak antigenically with some protein that will make the immune system kind of notice it a little bit better. All right. And it's kind of complicated. I was reading more about this before class because we have a lot of different kind of variations of this vaccine. The one that I mentioned before, I think the brand name is Prevnar 20. We see that on commercials. It's also called PCV 20, which stands for pneumococcal vaccine. Okay, again, I'm pretty sure those are the same thing. The 20 stands for the different variations of the capsule that are included in that vaccine. Okay, and this takes the different kind of polysaccharides in the capsule, and it's conjugated with a protein from the bacteria that causes diphtheria. It's a Corynebacterium diphtheriae, I think. So they've taken just a protein from that, again, to get the immune system to notice that a little bit. Okay. Again, this vaccine has a lot of different kind of permutations to it, like depending on the age of the person, whether you're a baby, whether your health status, if you're or if you're older. Again, there's different varieties of this. But we first came out with this vaccine. I don't remember if I posted this graph or not, but we first came out with this around the year 2000. And at that time, they called it PCV7. So they had 7 different variations of the capsule back then. And if you look at the incidence of invasive pneumococcal disease, so that's probably meningitis and pneumonia. I don't think ear infections are considered particularly invasive. You see the incidence went way down, cut in half. And then we kept getting better and better. Around 2010, we had 13 different varieties. And not only those, so this plots all invasive disease and just the ones that are covered by those 13 different kinds, and even the ones that aren't covered in the vaccine also decreased. And again, it's even better now because we have 20 different varieties that must have come out within the last few years. So again, it's really living, and it will limit the ear infections as well. Okay. All right, questions about Streptococcus. All right, so moving on to another gram-positive genus, a couple different gram-positive bacilli that I want to talk about. One particular in the Bacillus genus, and then we're going to talk about one of the Clostridium. So the Bacillus and the Clostridium are kind of, again, they're big gram-positive rods. The Bacillus are more aerobic and the Clostridium are more anaerobic. So Bacillus, we've used this in lab quite a bit because most species of Bacillus are pretty benign. They're not going to cause an infection. When you see those growing on a plate, they tend to form large colonies and they're kind of wrinkly, almost look like they're kind of erupting up sometimes. So sometimes we see that as a contaminant growing when we don't want it to grow, actually. But this genera of organisms, they're pretty much considered environmental. Like I said, they're not typically causing disease. They're in the soil, they're in the water. You probably have some on the bottom of your shoe right now. And like I said, mostly they're aerobes. They're considered saprophytic. There's a term we talked about a while ago. We talked about how most of the time fungi are saprophytic. All right, that means, anybody remember what it means? Yeah. Yes, so it feeds on decaying matter or dead, I'll say dead and decaying matter. Okay, they're decomposers. It's a soil organism, so some are going to be, there's a few that are sources of antibiotics. And the other famous thing about the Bacillus and the Clostridium both is that they form endospores. So those of you that are in lab, you got to start thinking about your lab exam next week. Remember when we do a Schaefer-Fulton spore stain, the vegetative cells stain red and those spores will show up as these small green ovals. Okay, and that's a nice dark one there. The ones that we did didn't really turn out quite that dark. Okay, so that where does the organism form endospores? Usually in the environment. Okay, there are a few species of Bacillus that do form infections. They're not going to sporulate inside our body. As a general rule, there's an exception to that, but as a general rule, they sporulate in the environment, where it's too cold or not enough oxygen or not enough nutrients, something like that. In our bodies, it's, a nice warm spot, access to a lot of nutrients. That's not when they're going to sporulate. Again, there's one exception. Some of them do form capsules. And again, they're going to encapsulate in a host. Okay, so they don't usually form capsules in the environment, spores in the environment, capsules in a host. As a general rule. Okay, so a few examples. Bacillus subtilis, again, that's one that we just kind of use in lab. That's not harmful at all. Bacillus cereus or B cereus. There's probably some joke in there that I haven't come up with it. But that one is most famous for causing food poisoning. And especially, they call it Chinese restaurant syndrome. So I enjoy a good Chinese buffet as much as anybody, but if rice has been contaminated with Bacillus cereus spores and it wasn't cooked for long enough, then when the rice is out on the buffet table, like warming for a long time, those spores can germinate. And then when we ingest the vegetative cells, that's when we get sick. It's not a particularly dangerous food poisoning, but you're pretty, feeling pretty lousy for a couple days. And then I want to spend a little bit more time on Bacillus anthracis because that's the causative agent of anthrax. And although anthrax is very rare, oops, come back. Sorry about that, press the wrong button. Okay, although anthrax is really rare in the US, I think, how rare is it, you should ask? I think there was only there was 25 cases from the year 1976 to 2001. So about one case a year. And then of course we had the 9/11, I think there was 22 cases in the US that year. So mostly we think about anthrax as, being something that we could get intentionally, and that's why we need to kind of be aware of it. worldwide, it's in the soil. So epidemiology, soil reservoir, there's about 2,000 cases per year worldwide, because in some areas of the world, it's prevalent in the soil. Middle East, there's anthrax spores in the soil. Not as much in the US, maybe like in the Dakotas a little bit more, but it just varies different parts of the world. And traditionally, anthrax has been a disease of farm animals. So we only get it when we're in close association with farm animals, especially hoofed animals, apparently. Okay, so remember, when something's zoonotic, that means it's spread between people and animals. It's important to understand that people don't give it to other people. Maybe there's some case where that's happened once, but you have to try really hard for a person with anthrax to actually infect another person with anthrax. So we'll say that rarely happens. Okay. The virulence factors, there's not a lot, kind of, you would think, for such an organism that's successful in killing a host so quickly. There's really not a lot of virulence factors. It does produce a capsule. Again, this is going to be once it's already in a host. And it's kind of a unique kind of capsule. Most of the time, capsules are made of polysaccharides, of sugars, repeating sugars. The anthrax capsule is made of poly-D glutamic acid. Okay, glutamate, that's amino acids. Okay, so kind of a amino acid, or more of a protein capsule. And that's still not very antigenic, because it's the same amino acid kind of repeated over and over again. So it's still, the immune system doesn't notice that very well. And you can kind of see that here. I'm not sure what kind of capsule stain this is, but you can kind of see the capsule there around the bacilli. And I should mention, if you gram stain anthrax, it has a very characteristic boxy shaped cell that you don't see anywhere else. So you can almost look at that and say, oh man, that looks like anthrax. Okay, chains of these rectangular cells. That capsule does, again, what capsules usually do. They resist complement and make it much harder for them to be phagocytized by our white blood cells. The organism will produce 3 different exotoxins. One exotoxin is just kind of a Trojan horse to get the other 2 into the cell. I think they talk a lot more about toxins if you take like medical bacteriology. But one toxin is called edema toxin, and it does just what it sounds like. It causes the swelling of tissues. Okay, so it actually upsets the water homeostasis in our tissues. And the lethal toxin does what it says it does. It causes cell death. It actually interferes with some of the signal mechanisms between cells, between host cells, and causes cell death, especially in the macrophages. Okay, so again, that's a really important cell. For losing our macrophages, then that pathogen can't be presented to the adaptive immunity, and really causes a wrench in our immune response. So a couple of reasons, not only for the toxins, but a couple of reasons that anthrax infection is interesting, because how the disease is manifested depends on the portal of entry. Okay, so whether you get it in a wound, or you inhale it, or you eat it, it makes an enormous difference in what the infection will look like. So if you had to get anthrax, the best kind to get is cutaneous anthrax, because It is the most common form, again, still pretty rare, but it's the least likely to kill you. So that's when you get the spores into some kind of a wound. All right, and that will cause a nodule to be formed in the wound. A nodule is just a abnormal aggregate of cells. I think there's two G's there in aggregates. Sorry, abnormal aggregate of cells. As that nodule persists, that that causes the tissue around there to die, and necrotic tissue often turns black. So this causes this black scab called an eschar. Got that formed over here. And that looks really painful, but apparently it's not. So sometimes, if you can believe it, sometimes people would just ignore that because it doesn't hurt. And actually, most of the time, it will heal up on its own. Okay, so mostly heals on its own. Occasionally, it will, those toxins will get into the bloodstream. And when it does, it can cause about a 20% mortality if it's not treated. So even then, at that point, if it's treated, then it's usually pretty treatable. So kind of surprisingly enough, again, this is the best way to get it through a wound. And I think those 9/11 attacks, 2001, there was 22 people that got anthrax, and 11 of them, it was cutaneous anthrax. So people that were in the mailroom and associated, with these letters that were mailed with Congress, or they were opening the letters. There's a really interesting, if you haven't seen it, a documentary on Netflix called Anthrax Attacks that kind of doesn't talk about the pathology of anthrax, but it talks about kind of those events and how they tried to figure out who did it, and a lot of stuff that I didn't know or I totally forgot about. So, and most of you weren't even born then, so it'd be interesting maybe for you to watch. Who's at risk of cutaneous anthrax? So again, typically people that are around animals. Okay. People. And again, not so much in the US because we vaccinate our animals. We have a good vaccine. And so again, it's really uncommon here. The kind that you don't want to get is the pulmonary anthrax, or sometimes it's called inhalation anthrax. That's when someone inhales the spores. Okay, so it's not the vegetative cells that are the worry here. It's when we inhale the spores. And 5 people died of this, again, back in 2001, where they inhaled the spores. It was kind of a whitish powder that was mailed to Congress. So once someone inhales the spores, oh, and I should mention that this is also referred to as wool sorter's disease. Because again, it was usually people that were around animals, or they were shearing sheep, for example. So the animal would be maybe coughing out some spores, and sheep shearing is a really dusty process. There have been people that have gotten inhalation anthrax just from the wool that was, shipped overseas, even infected wool. Okay, but so after one inhales those endospores, typically maybe after a couple days, they will start a prodromal phase. Remember, that's mild general symptoms, or they have flu-like symptoms. Okay, so a little achiness, maybe some fever. Probably the first few people that, when that showed up, they were probably the ones that died, because, there wasn't anything about that flu-like symptoms to make doctors think, oh, we need to treat you for anthrax, right? And then once the toxins, though, once the bacteria starts making those two toxins, edema toxin and lethal toxin, and gets in the bloodstream, then the patient usually has about 24 hours to live. Okay, so it's really fast acting. You know, animals that would get this would seem fine one day and then just drop dead the next. So again, within about, 24 hours, then we get this, prodromal phase maybe lasts for about 2 days. Again, and then once the toxins, they acts really, really quickly and 100% fatal without treatment. Even giving antibiotics once the toxins get into the bloodstream don't do any good. So now we have some antitoxin that we can give to neutralize the toxin. But again, I don't think that was available back in 2001. So what characterizes the disease? Really high fever, pulmonary edema. All right, so here's a chest x-ray. We refer to this as mediastinal widening. Okay, we get kind of the center of the thoracic cavity here looks wider because of the fluid buildup in the lungs. Patients have so much inflammation, they can go into shock. Those macrophages are being killed by the lethal toxin. And again, pretty deadly. Okay. The third way that anthrax can manifest, we call it gastrointestinal anthrax. And this is mostly the way animals would get it because, they're eating, say, grass in the pasture and they ingest some spores. Okay, so this is when spores are ingested. People can get anthrax this way if they eat some contaminated meat. So the farmer notices, oh, the sheep is sick, I'm going to slaughter it right away and sell the meat. And again, humans would eat it. So again, it's going into our digestive tract and it's going to cause necrosis and bleeding in the intestines. It also will cause abdominal swelling. So that's what ascites. That's ascites. That's a term for abdominal swelling. And I looked for a good picture of that and I couldn't find one. But okay, so this is again just as bad as pulmonary anthrax, near 100% mortality without treatment. There is actually a fourth way that's been described, and we'll call it. Where did I write this down? We'll call it injection anthrax. And there haven't been a lot of cases described of this, but this is mostly IV drug users where they have dirty needles or the drug is contaminated with anthrax spores. Causes we'll call it soft tissue damage. So it's a little bit more serious than cutaneous anthrax, but not nearly as serious as the pulmonary or the gastrointestinal anthrax. So little pussier wounds involving a little deeper tissue. Okay, so how do we treat anthrax? We give antibiotics. We won't get into the specific kinds there. There's some different ones that are commonly used. If we understand that you might have been exposed to spores, then anybody exposed would be given antibiotics prophylactically before they show any symptoms. Again, in 2012, we had some monoclonal antibodies approved that should neutralize the toxin. So we're a little bit more on our game now since those attacks in 2001. We do have a vaccine. We have a really good vaccine in animals. It works great. Again, they're not routinely vaccinated unless they're kind of known to have spores. Unfortunately, we don't have a good human vaccine. It's given in like 5 doses over a period of 18 months. Sometimes it's referred to as an acellular vaccine. Sometimes it's called a toxoid. Basically, it's part of the toxin. It's it's called the AVA, which stands for anthrax vaccine absorbed. And we're not giving, we don't give this just to everybody. It used to be, oh, we only give this to people in the military that might be going to the Middle East or where they might encounter it. But the problem with the vaccine is that about 1% of people that get it have some severe neurological reactions to it. So people in the military even started to be like, oh, we don't want to get this. So we don't even give it to the military anymore. It only works for a year, and then you have to have a booster every year. So it's kind of a lousy vaccine. All right, we'll call it good for today. We'll move on to one of the Clostridiums and tuberculosis next time. Have a good day.